In 1977, a group of scientists led by Nobel Prize winner Frederick Sanger published the first full genome, a virus called 𝗖cie RH174. It was 5,386 bases long and was published as plain text within the pages of the journal Nature. Fast forward to today, sequencing a human genome yields around 3 billion base pairs. If we attempted to store all that genomic data on paper or as simple text files, the sheer size would create overwhelming challenges for storage and transfer. To manage this massive amount of data digitally, file compression became essential. So how did we go from publishing genomes on paper to efficiently storing and analysing billions of base pairs digitally? As scientists began working with digital files, they initially stored DNA sequences as plain text, so just the letters A, T, C, and G in a file. But how does a computer actually store these letters? Computers only understand binary, ones and zeros, so each letter needs to be converted into binary code. Initially, scientists used a standard encoding system called 𝗖cie, which was originally designed for English text. In 𝗖cie, each letter takes up 1 byte of computer memory, which is 8 bits. Think of a bit as a single switch that can be either on, 1, or off, 0, and a byte as 8 of these switches lined up in a row. Here's what happens when we store DNA base letters using 𝗖cie. So our simple DNA sequence 𝗖𝗄𝐶𝗵 takes up 32 bits or 4 bytes of storage space. This is incredibly inefficient when you think about it. DNA only has 4 possible bases, A, T, C, and G, but we're using the 𝗖cie system designed to represent over 250 different characters, including uppercase and lowercase letters, numbers, and special symbols. Scientists realized they could create a much simpler encoding specifically designed for DNA. In this new encoding, we only need 2 bits to represent 4 different values. Now A becomes 00, T becomes 01, C becomes 10, and G becomes 11. With this 2-bit encoding, our sequence 𝗖𝗄𝐶𝗵 only needs 8 bits total instead of the 32 bits as seen with the 𝗖cie encoding. That's a 75% reduction in file size. This simple encoding trick makes an enormous difference at scale. A human genome contains roughly 3 billion base pairs. Stored as 𝗖cie text, that's about 3 gigabytes of data. But with a 2-bit encoding, we can shrink it down to just 750 megabytes. Still large, but much more manageable. But there's a catch. Real DNA data extends beyond the 4 standard bases. We often use N to denote unknown bases, or include quality scores to reflect our confidence in each base call. Additionally, certain file formats require extra detail, such as sequence names or annotations. As a result, compressing real-world genomic data demands more advanced and sophisticated techniques. While 2-bit encoding was a good start for the basic 𝗖𝗄𝐶𝗵 alphabet, scientists needed more efficient methods to handle the complexity of real genomic data. In 1952, MIT student David Huffman was taking a class in information theory when his professor gave the students a choice: either write a term paper or take a final exam. Huffman chose the paper, a decision that would revolutionize data compression. Huffman's algorithm wasn't originally designed for DNA. In fact, it predated our understanding of DNA structure. It was a general compression method that would only find its application in genomics decades later as sequencing technologies advanced. Let's see how Huffman coding works with DNA. His breakthrough was elegantly simple. What if we assign shorter codes to letters that appear more frequently? Think about Morse code. The letter E is just a single dot because it's so common in English. Huffman formalized this idea with what we now call Huffman coding. Here's how it works with DNA. First, we count how often each base appears in our sequence. Instead of giving each base the same 2-bit code, Huffman would assign shorter codes to the more frequent bases. Now, instead of each base taking exactly 2 bits, the length varies. A only needs 1 bit. If we encode our sequence with these codes, the average bits per base drops from 2 to just 1.7. That's another 15% reduction on top of our earlier savings. This variable length encoding approach is particularly valuable for handling those ambiguous bases we mentioned earlier. Since symbols like N for unknown bases typically appear much less frequently than the standard 𝗖𝗄𝐶𝗵, Huffman coding can assign them longer bit patterns without significantly impacting the overall file size. The same principle applies to quality scores. We can treat each base plus quality combination as a separate symbol using shorter codes for common high quality bases and longer codes for rarer low quality or ambiguous readings. What makes Huffman coding so brilliant is that it adapts to the actual content of your data. For example, in some DNA regions like promoters, certain bases appear much more frequently than others. Huffman coding would automatically optimize for this. But there's an interesting challenge with genomic data. Unlike human language, where some letters like E or T dominate, DNA bases tend to appear in roughly equal proportions across entire genomes, about 25% each for A, T, G, and C. This limits how much Huffman coding can compress whole genomes on its own, confining its use to specific genomic regions with skewed base composition like AT-rich promoters. However, DNA isn't just a random collection of letters. It contains structure, particularly repetitive patterns. In humans, certain sequences like AATAA appear millions of times throughout the genome. This is where Huffman coding falls short. It treats each base independently and cannot recognize these longer, repeated sequences. Scientists needed a fundamentally different approach, one that could identify and leverage these patterns. While Huffman was optimizing individual symbols, two Israeli computer scientists were developing something revolutionary. In 1977, coincidentally the same year Sanger published the first viral genome, Abraham Lempel and 𝗖𝗶𝘁𝗵𝘁 at the Technion Institute unraveled a completely different approach to compression. Instead of focusing on how frequent each symbol is, they asked, what if we looked for repeated patterns in the data? Their insight was profound. Most real-world data, whether text, images, or DNA, contains reoccurring sequences that could be referenced rather than repeated. Their algorithm, now known as 𝗖𝗁77, works by scanning through data and keeping track of what it's seen before. When it encounters a sequence that has appeared earlier, it replaces the repeat with just two numbers, how far back to look and how many characters to copy. Let's look at an example of how the Lempel-Ziv algorithm compresses the following sequence. We start by reading characters one by one, adding them to the output. When we've reached the second 𝗖𝗁𝐺, the algorithm recognizes it's a repeat, and instead of storing the final 𝗖𝗁𝐺 again, the algorithm replaces it with a simple instruction: go back 8 positions and copy 4 characters. These two numbers take up much less space than storing 𝗖𝗁𝐺 again. But the real power of Lempel-Ziv becomes apparent with longer repeats. The human genome is filled with repetitive elements. Consider the 𝗖 sequence, which is about 300 base pairs long and appears over 1 million times in our DNA. That's 300 million bases that can be compressed into simple references. For instance, if we've already seen an 𝗖 sequence at position 1000, when we encounter it again at position 50,000, instead of storing all 300 bases again, we simply write 49,300, meaning go back 49,000 positions and copy 300 characters. That's just two numbers replacing 300 letters. But Lempel and Ziv didn't stop there. They quickly improved their algorithm with 𝗖𝗁78. While 𝗖𝗁77 uses a sliding window to find repeated patterns, 𝗖𝗁78 builds a dictionary of patterns as it goes, making it more efficient for certain types of data. This became the foundation of many compression tools you probably use every day. Zip files, GIF images, and PDF documents all use variations of the Lempel-Ziv compression. What makes these algorithms so powerful for genomics is that they adapt to whatever patterns exist in the data. Unlike Huffman coding, which needs to know frequencies in advance, Lempel-Ziv algorithms learn patterns on the fly. This is perfect for DNA, where different regions have different kinds of repetition. The results were dramatic. Bacterial genomes that took 5 megabytes could now be stored in just 2 megabytes or less. And for human genomes, with their high repetition, the savings were even greater. But there was still a problem. Finding these repeats requires scanning through all the previous data, which gets slower as files get bigger. Imagine searching through billions of bases to find matches. Genomic scientists needed something even more specialized, an algorithm that could compress DNA while still allowing fast access to specific regions. By the early 1990s, genome sequencing was accelerating rapidly, and scientists needed more powerful compression techniques. In 1994, two computer scientists at Digital Equipment Corporation, Michael Burrows and David Wheeler, developed a transformation that would revolutionize both data compression and genomics. The Burrows-Wheeler transform, or BWT, isn't actually a compression algorithm itself. Instead, it's a clever rearrangement of data that makes compression dramatically more effective, and it achieves this using a surprisingly simple process based on sorting. Let's break down how the Burrows-Wheeler transform works with a simple DNA sequence: 𝗖𝗁𝐺T, 𝗖𝗁𝐺T. First, we create all possible rotations of our sequence by shifting one character at a time. Watch what happens on the screen. Next, we sort all these rotations alphabetically. Since DNA sequences use the letters A, C, G, and T, we'll sort them in that order. Now, for the transformation itself, we take just the last column of this sorted table. These last characters form our transformed sequence TTAACCGG. This is the Burrows-Wheeler transform output. But we need one more piece of information to make this reversible. We need to store the position of our original sequence in the sorted table. In this case, it's the first row, so we store the index 0. So why is this useful? Look at what happened to our sequence. The original 𝗖𝗁𝐺T has been transformed into TTAACCGG, where similar characters are now grouped together. We have 2 T's, then 2 A's, then 2 C's, and finally 2 G's. This grouping is the magic of BWT. It clusters similar contexts together. With this transformed sequence, we can now apply simple compression techniques. For example, run length encoding would replace these runs with a count. So TTAACCGG becomes T2A2C2G2. For longer genomic sequences with many repeats, these runs become much longer, creating excellent compression opportunities. So a sequence like this would transform to create even longer runs of similar characters. What's remarkable about the Burrows-Wheeler transform is that it's completely reversible. Even though the transform sequence looks scrambled, every bit of information needed to reconstruct the original is still there. Remember, Burrows-Wheeler transform itself isn't compression. It just rearranges data in a way that makes standard compression algorithms work exponentially better, which is perfect for handling the massive datasets in modern genomics. The impact of Burrows-Wheeler transform on genomics extended far beyond compression. By the mid-2000s, advancements in DNA sequencing technology led to faster and more affordable sequencing, ushering in the era of next-generation sequencing, NGS. This breakthrough allowed scientists to generate millions of short DNA reads at an unprecedented scale. However, this also introduced a new challenge, determining where each read originated from within the genome. To do this efficiently, researchers needed a method to rapidly map these short sequences to a reference genome, a crucial process known as alignment. This is where Ben Langmead and his colleagues at Johns Hopkins University had a breakthrough. They realized the Burrows-Wheeler transform could be combined with another technique called the FM index to create extraordinarily fast search algorithms. Their tool, called 𝗖𝗁, could align millions of DNA fragments to a reference genome at unprecedented speeds. Traditional alignment methods were painfully slow. They had to look through the entire 3 billion letter genome for each fragment. Imagine trying to find where a sentence belongs in a book by checking every single page. Burrows-Wheeler transform-based tools like 𝗖𝗁 and BWA, Burrows-Wheeler Aligner, could essentially jump straight to the potential matches, like having a magical index for the book, reducing alignment time from days to hours or even minutes. Let's see just how dramatic the difference was. This speed improvement wasn't just convenient, it fundamentally changed what was possible in genomics. Projects like the 1000 Genomes project, which aimed to sequence the DNA of thousands of people from around the world, would have taken years instead of months without these Burrows-Wheeler transform-based tools. And remember how we said that the Burrows-Wheeler transform itself isn't compression, but makes compression easier? When specialized genomic compression tools were applied after using the Burrows-Wheeler transform, the results were remarkable. By rearranging the data with Burrows-Wheeler transform first, these tools could achieve much better compression than was previously possible. This resulted in specialized genomic compression tools achieving 4 to 6-fold size reductions compared to the original sequence data. What began as an elegant mathematical transformation had become essential to modern genomic science. But despite the power of Burrows-Wheeler transform, genomics continued to evolve. By the 2010s, as sequencing costs plummeted, the challenge shifted from merely storing raw sequences to efficiently handling alignments and capturing differences across many similar genomes. This new era would require another leap in how we store and process genomic information. The Human Genome Project, which ran from 1990 to 2003, took 13 years and cost nearly $3 billion. But by 2014, advances in sequencing technology had brought that timeline down to just a few days at a cost of only a few $1,000s. This rapid progress created an enormous data storage challenge. Scientists at the Wellcome-Sanger Institute in Cambridge, UK, named after DNA sequencing pioneer Frederick Sanger, were facing this challenge head-on. They realized that traditional compression wasn't enough. They needed formats designed specifically for genomic data. The first breakthrough came with the Sequence Alignment Map format, SAM, which stored not just the DNA sequences, but information about how they aligned to a reference genome. Think of alignment like figuring out exactly where each piece of a DNA puzzle fits in the complete picture. But SAM files were plain text and enormous, so scientists quickly developed BAM, Binary Alignment Map, which was essentially the same information in a compact binary format with indexing. Indexing is just a system to quickly locate specific data. Let's look at what the BAM actually stores. When we sequence a genome today, we don't get one continuous string of DNA. Instead, we get millions of short reads, fragments of around 100 to 150 bases long that overlap. BAM stores these reads along with their positions on the reference genome, quality scores, and other metadata. BAM was a huge improvement, but Marcus Fritz and his team at the Sanger Institute had an even more radical idea. They asked, why store all the information when most of it is identical to the reference? This insight led to CRAM, Compressed Reference-Oriented Alignment Map. Instead of storing each read in full, CRAM primarily stores the differences from the reference genome. This is incredibly efficient because human genomes are 99.9% identical. Out of 3 billion bases, individuals typically only differ at a few million positions. Think of it like this. Imagine you have millions of nearly identical books that differ only by a few words on certain pages. Would you store complete copies of every book, or would you store one master copy and then just note the differences for each variant? CRAM takes the second approach. Let's see a concrete example. If we have a sequence read that perfectly matches the reference genome at position 1000, BAM would store the entire sequence along with its position. CRAM would essentially just store position 1000 matches reference for 12 bases. That's a dramatic space savings. And the space savings get even more impressive at scale. In the UK's 100,000 Genomes project, switching to CRAM reduced storage requirements by over 70%. Instead of needing 30 petabytes of storage, that's 30,000 terabytes or 30 million gigabytes, they can store the same information in less than 10 petabytes or 10 million gigabytes. But CRAM isn't perfect for every situation. It relies heavily on having a high quality reference genome, which works well for humans but can be challenging for less studied organisms. Also, highly divergent sequences like cancer genomes with massive rearrangements don't compress as efficiently because they differ more from the reference sequence. These specialized formats didn't just save space, they transformed genomic research. The combination of efficient storage and fast access meant scientists could work with thousands of genomes simultaneously, leading to discoveries about rare diseases, cancer, and human evolution that would have been impossible just years earlier. As sequencing technology continues to advance, bioinformaticians keep refining these approaches, finding new ways to balance storage efficiency with accessibility and analysis speed. Our journey through genomic data compression spans decades of innovation. From the earliest days of DNA sequencing to today's massive genomic databases, the challenge of storing genetic information has driven remarkable advances in computer science. Let's recap the milestones that brought us here. What started as a simple problem, how to store letters on a computer, evolved into sophisticated algorithms that capture the unique patterns of life's code. Each innovation built upon the last, combining insights from information theory, computer science, and biology. The impact extends far beyond just saving disk space. These compression techniques have enabled projects that were once unimaginable. The 1000 Genomes project catalogued human genetic diversity across populations. The Cancer Genome Atlas mapped the genetic changes in different cancers, and ancestry testing companies have helped millions explore their genetic heritage. Today, as sequencing costs continue to fall, we're entering an era where genomic data will become a routine part of healthcare and biological research. The algorithms that we've explored make it possible to store, transfer, and analyze this wealth of biological data. And as we stand at the threshold of new genomic frontiers, it's exciting to imagine what the future holds. Perhaps some of you watching will be a part of the next leap in compression technology. With the rise of AI, promising research is already underway. AI algorithms are being developed to identify even more subtle patterns in genomic data, leading to potentially greater compression ratios. If you found this journey through data compression interesting, then do like and subscribe as there's more content coming your way.